Okay, it's recording.
Okay, so today we're going to go through the pre-processing pipeline for whole brain imaging datasets that NAPE Center has been developing and distributing. The first thing we want to do is to check our access to the high performance computer cluster that we're going to be using. To do that, the first thing we want to go do is to go to this portal access, which we get credentials that we can use for getting allocations on the cluster. Let's actually go out. So once you log into Access, you can go to this My Projects page. If you have already submitted a project, you will see your new project here. If you're already in a project, you will also see it here. If it's your own project, you will be a PI or a co-PI, depending on how you made the project. If you are just a user, you will be listed here inside under the PI co-PI part here. So for now, I am a co-PI for this project OTH24003, which has a lot of credits. So here you go. So this is a page where you check how much credits you have and how much you have already allocated to use on specific clusters. You can also see it like here. Now you can see here that we are using PSC, Pittsburgh State University, Bridges 2, that's the name of the computer cluster, EM, extra memory node, and then RM, regular memory node. So we already have some resources or credits that's been allocated to those clusters. PSC also requires to get some storage space, which is called the PSC Ocean. So we have 5,850 credits used on the PSC Ocean. We have 165,000 credits on the regular memory node, 9,810 credits for the extra memory node. If you want to increase this number, what you can do here is say, if you want to add 20,000 more credits, you just change the number, put in a reason here, so additional credit for data processing, and submit for approval. Access will evaluate the usage or the change and then grant it or like reject it. And then once that's granted, you will see it reflected here. So if you're the first time users for PSC Bridges 2, you will also have to set up your credentials on their side. To do that, the first thing you want to do is to go to, you want to go to the web page. And then you can see viewer the Bridges 2 user guide. So this will have a lot of resources or documentations about their cluster. First thing you want to do is set your PCSC password here. There will be a link here where you can change your password. So click on this. And then here you can go through this password change utility to set up your password. Okay. So once you go through that, what you will have is an user ID that you got from Access. The same user ID will be used on any of the computer clusters you will be using through Access. So on PSC, I'll be using the same user ID as Access. And then you will use your password that you just set up here. Okay. So next you want to log into PSC. Oh, sorry, log into Bridges 2. To do that, you will use something called SSH client.
Just to clarify like a little bit, so the first website is mainly I guess these actions and checking this page out is mainly if you're managing um, the resources for a given project. Project is a research proposal that you submit. If you are using um, cluster resources that have already been set up by, let's say, NAPE Center or maybe your lab's analysis manager, then you won't really need to do the set up here. In order to do your analysis, you do need to go through that password reset that Ken mentioned, and then on a more daily basis, go through the procedures that he's about to go through now.
Yeah, thanks, Charles. The next thing we need to use is to log in to the cluster computer. To do that, you will be using something called a SSH client. There's a variety of software that enables you to do that. One common one will be PuTTY, so I'll be using that for today. So once you open up PuTTY, you will have something like this. This is the way you log into the PSC. So you're going to use a connection type SSH. So that's why this is called an SSH client. Each cluster computer will have their own host name. It's something similar to an IP address, but it's like it's something that specifies where you want to connect to. The host name for bridges to PSC will be this, bridges to PSC edu. Make sure that this is correct. And also, this is specific to bridges, so it's not going to be usable for like any other computer clusters. Use this and go to open. Then you will have this window. You start login from this window. You log in by first entering your ID, KISHII, that's my ID. And then this will next require your password. So you type in your password. And then once it goes through, you will get this screen. To make this bigger, you have connected to BR012 IB Bridges PSC. So this is going to be the login node of the Bridges 2. This is going to be where you're like working around first. If you see this, you're successful. You got into the cluster. On the same screen, you will see something like this. These like white shaded boxes. That is going to be referring to all the projects that you're participating in. So I am currently participating in 3 projects, bio 244 something, 240030P, and then OTH 240001P. So these IDs will be your project IDs, and then you will be using credits from specific projects if you specify which project you're going to be working on. Okay, so importantly, you just have to remember a specific project name that you're going to be working.
And this software basically allows you to communicate with the cluster in order to submit interactive jobs, if you will. Basically, that's just um, an equivalent would be to like remote into uh, a uh, a computer and to control it. And then also, once you do that, you'll be able to execute scripts and code.
Okay. So once you're in this SSH client, you've done one thing, right? Next, another thing that will be very useful is to have, is to have a software management system. I like to use this client called WinSCP. This is a very useful software management tool. As we did in PuTTY, you can also first log in to Bridges by using the host name and your username and your password.
Software and file management as well. I think the
Software
primary
and
thing is,
file name, yeah.
yeah, to be able to transfer files from your local computer to
Yes.
the cluster and vice versa.
Okay. To talk about WinSCP specifically, you will see a screen like this. Once you log in, so this indicates I'm logged into my Bridges account here. And then you will see 2 screens split it into half. In this left side, you will see your local file. So this is my desktop, and then I'm inside this folder named scripts. I'll be using all the files inside here later. On the right side, you will be seeing your structure of files inside the cluster computer. In Bridges or in any type of high performance cluster computer environment, most cases you will have your own home directory like this. So it says jet, home, and my user ID. So this is going to be my home directory. You won't have to work in this folder much. And what you will be more commonly be using is the data storage path that you purchase from PSC. So that will be located in somewhere else. And then first you want to go navigate yourself to there. The easiest way to navigate yourself is to by double clicking this part, you will get to this open directory path, and then you can type in the target path. The target path will be this ocean projects. Okay, so inside here, there will be a lot of projects that has been created inside PSC. And after that, you specify which project that you want to be working in. As I said, this is OTH240001P is my project ID. So I'll be using this. And then if you click OK, you will go into a data storage for the specific project.
You'll have to type that out because I think Ken's already bookmarked that specific folder and he
It's
can see it
very
down there.
handy if you have it bookmarked, but you can also type it in. Yes. So once you're inside, there's a lot of files here. So actually, these are all individual usernames. So Keishi, this is mine. Zjo12, this is Charles's, and there's a lot of other people's as well. There will also be a folder named shared, which you can store shared files that will be used across users. Each file will be have each file will be only accessible for specific people. For example, my folder is only accessible to me. I cannot go into Charles's file. So that's how you separate the credentials for each user. So for example, I cannot go into Charles's name. Permission denied. There you go. But you can go into this shared directory. Okay. So once you're in this folder, that's good. What you can typically do is upload all of your data in the shared directory and then access that to do your analysis. Here, I've already uploaded a lot of data onto the shared data path. So if we go into one of these, I will be today using this one. There's a long name, but it's called C8. So I'm going to use the C8 data. Inside here, there's 3 files that I've transferred onto the cluster from the imaging microscope computer. One file, EX488, that's going to be the autofluorescence channel. And then the EX639, that's going to be the signal channel. You will have, it's taking time to load.
I guess while it loads, just to clarify, should people make their own subfolder with their name in the data folder? Because I can imagine that.
So if that
Um,
data is only yours, if there's nobody else using those, you can actually put them in your own file. If that, for example, if one data is going to be analyzed across multiple people, you should put it in the shared file. And in that case, you don't need to make a subfolder. I don't recommend making a subfolder. Okay, so now you can see that there's a lot of files. These are all one image Z planes coming from the Ishii microscope. Another file that is very handy is this metadata file. This is taking a lot of metadata that was and was exported through the Smart SPIM system. If you don't have this, or if you don't use a Smart SPIM, that's fine. Okay. So now another file that is very important will be this one stored in here, regions annotations. All of the Brain Alice files will be stored in this file. The file name can be anything, it can be anywhere, but make sure you have those files on the cluster and then it's well labeled and you know where it is. We will be using this information later. For example, this YK stores a unified brain atlas provided from the Clearmap, and then the 3 typical files that we will be using is this Kim ref adult v1 brain, Kim ref adult FP labeled brain, and then this atlas info Kim ref FP base labeled CSV file. So make sure you have at least 3 of these files. Another file that is very important will be this SIF file. You can see this .sif file extension at the very back. This is called the Singularity container file, which we'll be using to run our analysis scripts inside. Again, the name can be anything. It could be anywhere as long as you can access it, but you will be needing it on the cluster. I have also some documentations how to download this from our shared directory. So you can use that to download this onto your cluster directly. Okay. So the other files that you will be needing will be the scripts to run the pre-processing files. So here in the left, I have all the scripts ready, already downloaded from the GitHub shared the GitHub page that we share scripts on. We will be uploading this onto the cluster, specifically into this folder in my path. So you can see Ishii scripts folder. I already have a folder for these files. I will be transferring files onto here. But before that, we want to adjust some of the parameters of these files so that it will be working on the cluster.
So where can users get those scripts files just to get started immediately?
So you can directly reach out to me, or you can go to this GitHub page that we share our Clearmap branch. Inside here, there will be a scripts file, which stores all of these like template scripts necessary for batch processing. So please, you can directly go here and then save or download each of these files.
So the files in here in the GitHub is exactly what you have on your
It's
local.
mostly exactly, yes.
Okay.
Of course, you have some of the parameters and some of the variables are different, but yes. Okay. So let's open up, first of all, this variable file. Actually, let's first of all change these files named batch submit jobs and batch submit detection validation jobs.
You might want
These,
to also, sorry to interrupt, just highlight any of the discrepancies between your local scripts versus the GitHub, just so that it'll people have like less errors that they'll encounter.
so the difference between the two in the local, it's actually mostly the same. They're inside the scripts that's on the GitHub. There's some parts that is not specified yet. You will see change the following depending on your file. You'll see a part here that you probably need to change. Same for this one, you have to change this part. This file, you don't need to change anything. This file, you don't need to change anything. So, sorry. This file, you don't need to change anything as well. The 4 files you need to change will be these files that will be ending with this pet.sh. These files you will need to change. Everything that you need to change will be commented like here. And then the other 2 files you will be needing to change is this variable file.xlx, this Excel file that you cannot open here, and then experiment parameter test. So this CSV file too. These 2 files will be specifying, one will be specifying which data set you're going to batch process. So if you have multiple data you want to process in a batch, you can just like list it into this file. Currently, this is like this has just some random things in it. Like it's like test1, condition1, and then file path is like some random thing, but you will have to adjust this depending on where it is on the cluster. For the variable file, let me open this actually here. So this is the local path where I store all the scripts. If you open the variable file, there will be a lot of parameters that you will need to change depending on your data sets. As default, we already have a well-established set of parameters for example, spot detection or like stack processing. So you might need to tweak it a bit depending on how it looks like, how the result looks like. but mostly the things that you will need to change is one here. So as I said earlier, you have to specify where your atlas files are. You have to specify that in this path, in this part of this Excel sheet. Inside the atlas parameter sheet, under this path red, XXX part. Okay. So let's do that then. So I am opening this variable file here, and we want to change things according to where the files that we want to use are on the cluster. The first thing I'm going to change is this, the atlas file, where the atlas files are. So we checked on the cluster, we have all our atlas files here in this ocean projects, OTH2 or blah, 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 shared regions, annotations, YK. So this, we need to copy this file path and write it here. If you double click on this tab here, you will get this copy path to clipboard. If you click, sorry, let me do this again. If you click this, you can copy paste it here. So now this is the path you want to, where you store all your files. So that's
A right
good.
click or is that double click?
So copy path to clipboard, you click it, and then you just do Control Paste. And then here you can also specify what the atlas file name, what the annotation file name is, what the atlas info file name is. If you're gonna change that, you can just change it as like this. So you get the name of the file that you want to use, you copy it here. Let me just make it easy. If it's like this, you can copy it here, paste it here. If yeah, so again, you can copy paste it here, and then so on. Okay. So now the variables for the aliases are set.
So one important thing to note here is that
Mhm.
um, this variable file will be read in by the main analysis script, and um, these parameters will be used by the main analysis script to refer to the atlas files. Hence why Ken is um, taking the paths from the right window in the file transfer program, because in the right window, those are the files that are on the cluster. And ultimately, all the analysis and file referencing will be based on the cluster storage system.
Okay. The other parameters that you will need to adjust. I think that will be, so the next thing you want to check, not necessarily adjust, is the crop generating parameter. I will explain later, but we will go through a process where we generate small crops of your entire image and do the cell detection algorithm onto these small crops. That will allow you to validate how well the cell segmentation worked. You can specify how many images that you want to crop and whether you want to run the detection algorithm on them or not. Those will be specified here. You can see the number of crops, and then if you want to split those images into some as a test proportion and some as a training proportion, you can specify that proportion here. Z offset will be specifying how many images that you want to ignore. If the offset is set to 600, that means that all the images from 1 to 600 will not be used to crop images, and you will start making images from 601. The crop size will mean how big their final cropped images wants to be, and crop per stack will be during this process, we will take a random Z position and then make 3 crops from there and then move on. Okay. Resampling parameter, please use a number that is exact same of the amount of cores that your node will have. This will be 120, so please use 128 for Bridge's regular memory node. And something like 48 or 96 for the extra memory node. Mainly, you just don't have to change this. Stack processing parameter, this will define how well you will do a thing called parallel processing. For regular memory node users, don't change these variables. Voxel parameter, this is something that will be more important when you do a spatial analysis or the box size analysis. Typically, we use something called a spherical method, and then we create a voxel of 10 micron to 10 micron to 10 micron. And then spot detection algorithm is parameters is consists a lot of parameters. It is more informative if you go to the Clearmap documentations, actually. So if you go to this web page where it documents how Clearmap is running, there will be a part here which specifies a lot of the details of these parameters. It will be better to read these documentations when you want to adjust it. And then the threshold parameter is used after you do the initial cell detection. If you think some of the detection is capturing cells that are too big or cells that are too dim or too bright, you can use this part to threshold them. Again, this will be this will be more well documented on this tutorial. So here, so in this algorithm, there will be two things that you want to specify, a row and then a value. Each row will specify a specific type of value coming from the cell detection algorithm. For example, row 0 will intend the intensity of the cell center in the raw data. 2 will be max intensity of the cell center after the background subtraction. And then 3 will be cell size and voxels after the watershed detection. What that means, if you want to threshold by like something like intensity after background, you will use 2 in row, and then the threshold will indicate that you're cutting off any intensity under 500 and over 35,000 here. If you want to add a threshold for size, you can do something like 3 and do like anything that's smaller than 1000 pixels will be eliminated, and anything under over 10,000 pixels will be eliminated as well. Okay. So once you set up all the variables in this variable file, you can save it and then close it. Okay. So the next file you want to adjust will be the experiment parameter file test. So this CSV file, as I said, will be saving all the data that you want to process in batch. Today, I'll be only having one file here, but you can have something like a list of like data sets that you want to process in batch. So the important information that will be used here will be, first of all, this one. If you have collected your images on the SmartSpin 2 microscope, if you specify that here, it will use a specific way to calculate extract the metadata out of the metadata file. So if you have if you have collected images using the SmartSpin, just specify SmartSpin 2 here. The base directory will be referring to where the data will be stored. So again, you go to your file managing software, and then you navigate yourself to where the file will be. So in this case, the data set that I want to process will be C8, and then it will be located here inside the cluster. So as we did when we were getting the Alice information, go to here, right click, and copy path to clipboard, go to your Excel sheet, and then change this variable here. So now we have the file path where the dataset you want to process will be located. And again, this will be the path inside the cluster, not on your local environment. we want to also specify where the variable file will be. So let's just finish this. So in my case, I will be saving all my scripts onto the folder here, inside my folder, inside this scripts folder. I will be transferring this variable file here, and this will be the path I want to specify. This will be with the file that I want to use for the data processing. So what you can do here is again the same thing. Actually, here you can do copy to clipboard, include paths here, and then go here and then change this variable here. Now this cell is referring to where the variable file is. Okay.
Just to be clear, that path is now pointing to the variable file that can transfer it over from his local computer to the cluster data storage location in the scripts folder. I actually have a question here. So this is just one session. If you have multiple, will the main script be generating a new node? Or ask for a new node to analyze the second data set?
Yes, so I will go to that part later, but the basic idea is that um, so the way the cluster computer works is that you request for a node and work a specific job on that node. The way our batch script work is that each of the data sets specified in this file, for example, this row of data set will be requesting one node. And then if you have a list of data sets, each data set will be requesting their own node. So memories and cores will be specific to the node type that you will be requesting. If you are going to be requesting a regular memory node, use the 240 gigabytes as memory and 128 as cores. The signal channel and the background channel is going to be specifying which image channel will be used as a signal or the background. As we explained, the EX4088 channel will be used as background, so I'm going to use that here. And then the EX639, the far red channel will be used as a signal, so I'm going to specify that here. The orientation of the image is going to specify how you want to reorient your image so that it will fit the brain outlets. The way I took the image for this data set is that I have the ventral side facing upwards. So I started the images from the ventral side and it went to the dorsal side. On top of that, it was a horizontal plane. So the y-axis and the z-axis has to be flipped in order to make it into a coronal section. So first if the way you think about this is that 1 will be referring to the x-axis, 2 will be referring to the y-axis, and 3 will be referring to the z-axis. And then the position in this bracket, so the first position, the second position, and the third position, is referring to what axis should be used for the final orientation. So swapping the 3 and 2 means that you're swapping the y-axis and the z-axis. And putting a minus on top of that means that you're going to flip the order of that axis. So in my case, the z position was starting from the ventral to the dorsal. So you want to flip that. So you have to put a minus on top in front of the 3. And then because we're going to flip the y-axis and the z-axis, we're going to flip the 2 and the -3 position into this order. So at the very end, we get 1, -3, 2.
So what's the typical final orientation?
It's either 1, 3, 2, or 1, -3, 2, or 1, -3, -2, or 1, -3, 1, 3, -2. It could be anything, to be fair.
So
So if you
when
okay, so if you have
okay.
the anterior part facing upward, you don't have to touch the 2 part. If you have the anterior part facing downside, you will have to flip it, so it'll be -2.
What would it be if, you know how we usually orient the brain in the machine, where the anterior part of the brain faces the user, and then dorsal faces up, and then ventral faces down? Would it
So
be
that will
what?
be 1, 3, -2.
Okay, cool. That's what I thought. Thanks.
The image processing method can be specified here. If you go into the Clearmap documentation, you will see that there's 2 methods. We will be using the spot detection method, so you can list it here. You can specify that here. XY resolution and Z resolution. This will be automatically captured from the metadata, so you can put this false, but if you want to specify that, you can also type in something like 2.0 micron per pixel, or like 8 micron per pixel. If you're using the smart SPIM and you have specified it, so here, you shouldn't worry about this.
Do you
Signal
have any models for um, projection, or is it only spot detection right now that's working?
spot detection, no, so that's a totally different. So anything projection will go use something totally different.
Okay, cool.
It's not on the same package. Signal file range will specify is specific to the cell detection part. If you don't want to do cell detection on your full data set, you can actually trim it down to specifics. So if, for example, if you do 600 to 800, what this will mean is that you're gonna do cell detection only on Z-plane 600 to Z-plane 800. So that will like avoid a lot of the processing time. Okay. So you get this. So now this experiment parameter tests CSV file is good. So you can start transferring this here too. Okay. Okay, now let's take care of these .py files. These .py files can also just go on to the cluster directly. You don't have to modify anything on these .py files. For these two, for these 2 sh files, you want to edit it a bit. So I will be opening these two. I will be using this text editor here. I like to use VS Code, so that's what I'm going to use. And everything that you need to change will be under this first path. The first thing you want to change is the experiment file, and that will be the name of this. So what we're going to be using experiment parameter file, experiment parameter has NAPE workshop. Put the file name here. We change it here. We can also change it here too. So the 2 files. The next thing we want to change is the next thing we want to get is the SIF file. So on this cluster, we stored it here in the shared path. So let's get the full path to this file, copy to clipboard, include paths, get this one, and change this to here, .sif. And then the time will be restricting how long you will make an allocation on the node. Make this like long enough so your code will finish by then. Usually the batch submit jobs like 4 hours or 6 hours will be fine. And then for the detection validation, 2 hours should be fine. You get these. Change the partition name to RM or EM. Typically use a regular memory, so RM. Change the allocation name to your project name. Here, change the same. And then the way the script works is that once the job is done, they will send you an email. So you can specify your email address here, and this will be used just when sending the email. Okay. When saving this, you need to use select end of line sequence to LF. So make sure this is LF. Okay. So, and then you can do file save, and then close. And then you can transfer these scripts to your path. And then here, okay. So next, open PuTTY again. I closed PuTTY, so let me open it again. The first thing you want to do is move to the path where you hold your scripts in. So you want to execute all your scripts while you're inside that folder. To do that, you use a command called cd. This stands for change directory. So get the copy the path of the scripts folder that you're going to use. Go to PuTTY, cd, and then right click. And then you can get this, and then you're moving into here. The next command you're going to use is the bash command. This will be executing all of these batch scripts. The first thing you want to use is this one. So batch submit detection validation jobs. So again, you got this, and then you can execute like that. So once you enter that, you will get something like this. The file name will be C8, the variable file will be this. You will be using this much memory, this much core, and then this tells you that you've correctly submitted the batch job. Okay, so when SAP is like re-updating what is inside the file, so if you refresh here by using this like green button, you can see some files that has been generated. Okay, if you sort it by when it was changed, you will see three new new files. The first one will be this job segval c8.sh. This will be the job script that was created and submitted. For each data set, you will have one job file. And then you will have two more files that is going to record what was happening during the process. This processing job will be, like, for example, when you run something, you get all of these like outputs. Those will be recorded inside there. And then the other thing that will be recorded is that .error file, ERR file. If there's an error happening, it will also be recorded here. What you want to do is once you submit all the jobs, you want to also use a command that lets you evaluate how your job is running. Oops. That command will be this. There will be a list of commands that's going to be very useful. These are called SERM commands, but one very useful command will be this. This is very long. You should have access to one of the documentations or like a PowerPoint, and you can probably just copy paste this. But it starts from SQUEUE, and then -U stands for the username, and then you put your username here. And if you copy paste this into PuTTY, you get something like this. This is specifying the job ID, this one, which the user me has been initiated, is still running. It has 2 minutes and 15 seconds passed. It's still running. And then it's just showing that it's currently running.
You want to show, I mean, I don't think we should cancel this right now, but just in case people realize that maybe they made a mistake in their parameters or they just need to cancel it, how, like, one would cancel an existing job.
So the way you can cancel things is you can use the cancel command. So again, oh, it doesn't say it here. Yes, the scancel command you can use. One way you can do it is you can specify the job ID here. So if you type in scancel 243084887 and press enter, you will be canceling this job. if you want to cancel all of the jobs that you submitted, you can do something like you hit Ishii, and all of the jobs that this user submitted will be canceled. Yep, so currently it should be running. Okay, so it's done. Okay, so now we can see that the file size has changed. And if you look into it, you see a lot of like these logs. This tells you that the cell detection algorithm was working and put in some information as an output. At the very end, you can see one file process ended in 230 seconds. The entire process ended in 230 seconds. So like roughly 4 minutes to just process the entire thing. You can confirm that by going to the data path. If you go here and refresh again, you see this new file crops. And then you will see like train and test. And then you see a lot of files that was generated. So what you can do is download these files. It will take a bit. So now you have these files here. And then you can open image.j. Image.j. Oops. Image.j. And then, actually, let's go to this file. Sorry. There we go. Crops, train, and you have a bunch of images. What this is that there's a list of files that was generated during the crops. This one, for example, is one cropped images, and then there will be a list of files that are associated to that. So if you open the cropped images in image.j, it will look like this, and you see an example CFOS cropped image here, right? And then the result of the cell segmentation will be stored in multiple files, but mainly this spot cells check file here. So if you uploaded this, oops, sometimes it doesn't load. In that case, what you want to do is import and bioformats, and then you can load the same file like this. What this gives you is the same image like this, but it will also have two channels. The green channel listed here will be showing you the spots where the algorithm detected the cell. If you adjust the threshold a bit or color balance a bit, if you adjust it to red, you can start to see the green dots are appearing on top of the cells that you think that there should be. Okay. You can also make something like a Z project. So this will also be showing where the cells were and then where the cell, like the coordinate of the cell was detected. That's green.
So
Okay.
does the threshold parameters in the variables file alter these results a lot?
Yes, so the two main files that you want to go through is the variable file that stores the parameters for the cell detection, but also the thresholding parameters, because some of these cells might not be picked up because the thresholding was too strong or or there could be some noise that is kind of getting picked up because the thresholding is too not enough. So yes, the main files, the main parameters that you want to adjust will be those two. So in another sense, you go to the variable files, and then you can go to the threshold parameter. So you can adjust this to make it work. Okay. So you will have, the cropping will not be perfect. It's not you don't necessarily have a lot of good cells in there. But yeah, you will have like enough. You can make a bunch of crops to um, make you will get a bunch of crops that should have some good cells in it, and then determine whether the cell detection algorithm worked well by looking into the cells checked file here. You can also feed the same detection algorithm with the cropped images that you manually use. So for example, if you have a good set of cropped images, you can go here and just place the files in this folder. Um, so say you want to test something here. What you will do is make a new folder with this name, make a change, make a test, and then transfer those cropped images here. For example, if this is one of your good cropped image, you can transfer it here. And then you can use the same algorithm using these images. To do that, one thing you will need to change is, if you go back to this variable file, in this crop generating parameter, there will be these two flags, a crop flag and detection flag. If you only want to do the detection part, you can put this to false. And then you can skip the crop part and then only use the detection. So if you have a case where you make your own manually cropped images and upload it onto a cluster or into a folder, and you only want to apply the detection to those images, you can do something like this. On the other hand, if you only want to do the crop but just not do the detection, you can do crop flag true and detection flag false. That will only create a lot of crop images but don't do the detection. Save this, here we go. Okay. So next thing is that once you set up all the parameters and once you confirm that the cell detection algorithm works well here, and you want to apply that onto your full data set, you go to the second script. So the second script will be this bash submit jobs.sh script. So the next thing you will do is submit that bash job. For this experiment, so for this demonstration purpose, I will modify one thing. So please open, I will be opening this file, and I will be specifying the Z range to only 400 to 420, because I don't want to go through the entire file. I'm just gonna only specify to a 20 images stack. Now you don't need to do this usually, but this is just for demonstration purposes. And save this, and then upload this here again. Okay. So now everything's set. So you want to use this script. Type in bash again and copy batch submit jobs.sh, and then you submit it. Now this job was submitted again. You get the same log here. And if you update this, you get this job ch.sh script right here, sh file here. So currently the status of the job is in, oh, it's running. Okay. So now you get the new files again. So now it's just job ch, processing job c8. It's different from the previous one. This was job segmentation validation segval file. So you can separate these two. Currently, the files is going through a very, very dramatic resampling and reorientation process. It will take probably like 5-10 minutes, I think.
So I just cut this part out.
Yeah, I'll probably just cut this part out. Um, okay, actually, this is good. So now we have some images um, created. So it's continuously making new images and exporting it. So if you download this, sorry, Charles, there's going to be a bit of editing probably
That's no
necessary.
problem. That's not too bad.
So the download is almost done. Okay, so you will get a list of images, but I'm just going to show you two examples here. One example will be after the resampling part. So if you remember the reorientations and things like that, first the entire Z stack of images will be resampled and reoriented by the way that you define in the variable file. The result will be looking like this. So this is a resampled image of the signal channel, the EX639 channel. If you load it into ImageJ, you can see that it was, let's just adjust the thresholds of it. You can see signal image is looking like this. So it's now from the most anterior to the posterior. It's in coronal. It's also not flipped in the y-axis. So the ventral is the bottom and the dorsal is the top. So you can see all the reorientation was correct. And then the resampling is correct too. You will have an image for that for the autofluorescence channel. So this will be for the autofluorescence channel. This will be for the signal channel. You'll also have a smaller version of that. So this file. This will be used to align the brain onto the atlas. So you have something like this. So let's go back to the scripts folder. There you go. So this is going through a lot of processing right now. There you go. So what is stuck right now, what it's doing right now is it's trying to align the this autofluorescence resample image onto the brain atlas. And that takes roughly 5-10 minutes. That's probably the longest part if you ignore the cell detection part, but that is taking some while. So yeah, let's just stop here then. Or do you want to go to like a already done one?
Mm, hmm. Wonder if they'll be confused. As long as the structure looks similar.
Let's just wait a bit then. Still gonna take a bit though. Maybe it was just better to uh, just start explaining everything after everything was done.
I mean, that's fine. Another thing that, so wait, sorry, I kind of might have missed this, but did you talk about how to set which channel is the autofluorescence?
Yes, I did.
Okay, cool.
I think, Charles, one thing that would be, please cut this part off, but one thing that would be useful is to, um, the workshop part, like the first part of the workshop, I think we can trim a video to like, just use that, and then swap the latter part of that workshop to this video.
Oh, so like
The
you're talking
work,
about it with the next time we do it?
No, so if you were to upload videos for like the general usage, we can use the first part of the workshop for like a primer, and then this video for like the hands-on training.
Yeah, yeah. Like you're talking about your lecture part. Your lecture part
Yes,
was pretty good. Yeah.
yeah. I think the hands-on part was just like a mess. I don't think people can recall get anything
Yeah.
out of it. Like I was not on my screen for like half of the time too.
Yeah, yeah. I think it'll have to do a lot of major editing. So I
Mhm.
think, I mean,
This
thus far, this one that you're doing right now, it's really nice.
yes. So let's just use this one then. And because like yeah.
Yeah, I think with this video as well as the PowerPoint that goes along with this, hopefully that gives people a good starting point.
Yeah, I agree. I think that would be a lot better than like the videos of the workshop for sure. Like,
I just hope that I wonder what kind of errors they people might come that might arise, and I hope that they don't have to bother you too much.
well, like if you have enough of the errors, we can make an error. What to do when this error kind of thing.
Oh yeah, that's true. Yeah, we can do like a follow-up
Yeah,
video. Much more is there after it finishes? I guess you'll just talk about the files.
yeah. No, actually, let's just skip it. I have a new. I have like the results file anyway. So
Okay.
let's just cancel this job. Okay. Then so just fast forward. And then we can go to data. I can populate this with the actual data. And then I can say like download it. Just like copying data onto local might just take some time too though. This. It's a lot actually.
Is it okay if I go to the bathroom?
Sure, sure.
I'll be back. Still waiting?
Yeah, give me 2 more minutes.
Okay, sounds good.
So I'll just explain that this is what it will look like on the right, and then I've already transferred the data onto my local on the left. So it's like a post time skip.
Sorry, say that again. I was messaging something.
Yeah, I'll just explain that the files will look like the right, and then it was already downloaded to the left.
Oh, okay.
Thirty, 90, 27, 26. Okay. Yeah. So is it okay?
Yeah, go ahead.
Okay. So once all the once the job is done, you will end up something that looks like the right. You have a lot more image files than when we started. So there's 3 files here. There's 3 new folders here, and there's a bunch of like new CSV files. There's a bunch of file names that ends with the NPY. These are called NumPy files. This is a list of like um, this is an array file that is used in Python. And then there's also 2 CSV files here. These folders are storing the transformation matrix that was used to align the brain atlas and the image. And then these CSV files are containing cell counts per region. So for example, if we open this, so I have the folder up. I have all of these files were transferred to my local here. And then I'm opening the folder here. And if you look at, for example, this annotated counts clean file, you will have something that will look like this. So this will resemble your brain atlas and info file. You will have the ID for each of these brain regions, and then you have these 2 count columns. The column that you should be using is this new count columns. This is counting all the cells within this region. For example, the motor cortex will be having 5,795 cells. I don't know. Primary auditory cortex layer 4 will having 93 cells, and so on. Okay. The list of TIFF images, some I've already explained, but the important ones will be 2. The first one will be this aligned atlas file. This actually is using the original atlas MRI file, and it's warping it so it matches your raw data. The file that will match this will be this autofluorescence resampled image. So if you also load this onto ImageJ, you will have 2 files that have the exact same shape, 361 Z-planes, 765 to 377. So if you overlap these 2 images with an ImageJ function in red and green, you will have something like this, which is showing the overlap of the 2 red images and the green images. Like that. Okay. Another image file that is important will be the cells heat map file. This is a result of all the cell segmentations, and all the cells are voxelized and mapped onto the brain atlas. So this is a good way to look at the cell distribution on the raw atlas plane. You can see where there were more cells and where there were fewer cells. Okay. The NPY files, you probably don't need to touch them, but these files will store the cell coordinates as well as something like intensity or cell sizes. If you load this NPY file in Python, you will have a very long array with a lot of rows. The rows will indicate one cell, and then they will have a column for like X, Y, Z, or like cell size, cell intensity, and so on. Okay, so those are the file structures after you finish this pre-processing pipeline. And then once you collect all of this data for all of your individual data sets, you can start proceeding to the data analysis step. Okay, I think that's it.
Nice. That was good. I think that was, yeah, it was really um
A lot more better. Yeah.
well, yeah, I think it's just with everything that I can if I stitch everything together, it'll be pretty seamless.
Yeah. Okay. So thank you for the recording, sticking onto recording. I think it will be like under like around that hour thing if you stitch out everything. Like yeah, about an hour.
Yeah, I think so. I'm gonna
Um
stop the recording.
yeah, I'll stop the